Okay, I was looking at the chat messages and I was seeing people talking about whether they need to go to the lab tomorrow. That's if you're enrolled in the 6019 section. That's when the lab is for that section. If you were in lab yesterday, 6017, you do not need to come to school tomorrow. Not for me anyway. So if there's any question about that, again, you can ask me here or we can talk about it later. Okay, otherwise, let us take a look at a short movie. And this is a nice one. I think you'll like it. So, Inner Life of a Cell. This is one that was done at Harvard University, which is one of our top schools, really one of the top schools in the world. And they did something where the animation is way better than in the one that I showed you. That was off this DVD, off this one. This Harvard one was done more recently, and it's got much nicer animation, and it plays kind of as a music video, just showing some of the beauty and complexity of what's going on in a living cell. I would like you to see this. Don't expect to understand it all. In fact, I've seen it many times, and I don't understand all of what's going on in there. I know what some of it is, but mainly, I just want you to see it to get a sense of kind of what's going on in cells. And so let us take a look at this. I will give you a link to this, so anyone wanting to look at it later could. Actually, I'm not sure if I was sharing that to you. Were you seeing the video?
No.
Yeah, sorry about that. Let's try that again. Okay, now you're probably seeing something like we're talking about.
There it is. That's it.
Good. Yeah, sorry about that. Okay, let's try it again. This only goes about 3 minutes.
Who had been working with animal tissues, noted that all animals are composed of cells. A decade later, a German physician, Robert Virchow, tried to understand how cells grow and reproduce. He concluded that existing cells give rise to new cells. Together, the ideas of
Okay, that's not going to be on the test. Don't worry about that. They have the version that I just showed you with no narration. There's also a longer one that runs 8 minutes where they actually explain what's going on. I think I'll put links to both versions in the week 1 folder here in Canvas. So anyone wanting to go and find out what was actually happening with that, the longer version will explain it. But I think in some ways it's nice just to watch it with the music and not worry about exactly what's happening there. But I hope that gives you at least a little sense of the beauty and complexity of what's actually going on in cells. That's why I wanted to show that. So this is what I have for us right now on the cell chapter, chapter 4. Again, there is a later section of that we will come back to in about 2 weeks. But right now, that's as much as we need to be doing in our chapter 4. I am going to be going back to try to finish what we have on our chapter 1 today. And again, I think we'll get out a little bit early. I'd like to know again if you have questions for me before we change back to the chapter 1. Okay, then let's do that. So back to chapter 1. So we had been looking at science. For instance, we had the situation with the childbed fever that was ravaging maternity wards in hospitals in the 1800s. Well, we mentioned that the work that the Semmelweis came up with here did not take the whole world by storm. Basically, his prescription about hand washing and sanitation did not catch on generally, not for quite a while, for various reasons. But at least he's on a stamp today in Germany. That's what this is. So, and what we do now with sanitation around hospitals, a lot of that comes from his work. So he definitely played an important role in us understanding the need to go and sterilize things constantly. So that is something where if you're involved in medicine, of course, you are obsessive about hand washing and gloving up and so on. And a lot of the reason for that goes back to the work he did. So, shifting gears here some, we have this fact that life forms are not isolated from the environments. Remember, life forms need to be able to absorb resources and also get rid of waste products. So they are tied to the non-living environments. They can't really exist separate from that. So they will take in, for instance, food, oxygen, water from their surroundings, and they will get rid of the breakdown products, the waste products, to the environments. And if you ever doubt this idea that you can't live in isolation, just put a plastic bag over your head for a few minutes and tell me what that feels like. You have to be connected to the non-living environment. And then the life forms themselves are part of larger ecological relationships. They get eaten by other things, they get broken down by other life forms. So even in our case, if we live to old age and manage to avoid getting eaten by something, bacteria will still break us down in the end. So we're part of larger food webs and food chains. And what happens is that your chemicals get recycled the atoms, the elements that we're made out of, those all get reused. Those atoms don't change. They will just change their arrangements with other atoms. Atoms will switch partners with other atoms, but the atoms themselves go on existing basically forever. But the energy does not. Energy flows through and constantly needs to be replaced. What is the basic source of the energy that we have for this planet? Where does our basic supply of energy come from?
The sun.
Yeah, the sun. And if the sun went out, well, we'd really be out of luck. We can't just keep recycling the same energy. Energy goes from being more highly organized to more disorganized. We need to constantly have a resupply of the highly organized kind of energy, which in our case is the sunlight. Otherwise, energy always degrades toward just randomness, which is what we call heat. Heat is just a random, disorganized kind of energy, and you need to replace the waste heat with more high quality energy. And as a result, we need the sun to keep on shining. Obviously, we get seasons where their life forms may hibernate and so forth and come back the next spring. But in general, we need the sun to keep on shining. So the atoms that are in us are essentially eternal, but the energy is not. But here's an example of a very tiny little ecosystem. We have sun coming in. You can tell because it looks so bright. And these plants are capturing that sun's energy. They're growing. Then these caterpillars are feeding on the plants and so forth. So all part of a tiny little ecological relationship there. Well, then we've got the different cell types. And I want us to look some at diversity here with life forms, keeping in mind, again, this difference between the big cells and the little ones. Complex, large, eukaryotic cells, less complex, simple, small, prokaryotic cells. Well, when we look at the diversity of life, we can break it all up into three of these categories, which are known as domains today. You take all the life forms on the planet, everything that's made out of cells, and we're able to essentially divide it up into one of three different categories, three different domains. Those domains are mostly distinct from each other with long evolutionary histories of their own, not totally separate from each other because we think there's been some crossing over between those. For instance, the mitochondria that our cells have started out as bacteria, and bacteria are organisms from a different domain, but for the most part, each domain has its own independent evolutionary history. And two of those domains involve prokaryotic cells. We have the bacteria and the archaea. Now, bacteria I've already referred to quite a bit. Archaea, you may not be familiar with that term, but think about bacteria-like organisms, but with some important differences from bacteria. In both cases, we are talking here about prokaryotic life forms. That means very small cells without a nucleus. But it turns out there's two styles of those out there. One that would fit into domain bacteria, the other domain archaea. Both of them involve small, simple cells. And then the third domain, that's our home domain, Eukarya. And that includes everything that's eukaryotic. That means plants, animals, fungi, all types of algae, and a lot of other kinds of life forms that fit in under this heading. So altogether, we've got these three different domains. They're all roughly equally distinct. Each of them is roughly equally distinct from the other two. So they've had very long independent evolutionary histories, making them very different from each other, even though the bacteria and archaea superficially look about the same. As we study them, we find they're not. Now, Eukarya, that has basically all of the familiar life forms in it. If you just look out the window and you look at life forms out there, everything you're going to notice will be in this category. As to bacteria, domain bacteria, well, here's what representative bacteria look like. A lot of them are spherical, known as cocci. That's a reference to the spherical shape. A lot of others are rod-shaped, bacilli, and that's what we're getting right here. Some of them even make a very thin corkscrew, spiral shape, and these are all extremely small, much smaller than a eukaryotic cell. Now, if you're doing microbiology later, you will study these a lot, and you'll be able to identify, for instance, cocci under the microscope. So that is something that could be in the future for you, 2 or so quarters from now. But these are typical bacteria. Here's a little more variation on their shapes, but you notice they never get to be very complex looking. They'll sometimes have chains, sometimes clusters of them together, like here. These are staph, and these are basically in clusters like grapes. So that's one distinguishing type of arrangement with the bacteria. But again, it's just clusters of bacteria together. Nothing more complicated than that. The archaeans, they look a lot like bacteria. Here at the left, this is extremely highly magnified. So this is super highly magnified at the left, where we've got clusters of these small archaean cells. Here at the right, this is where we have colonies of them growing on a petri dish. So each of these dots has millions or billions of identical cells in it. And these often do have kind of a pink or reddish look to them. But these are unusual because a lot of them they gravitate toward extremely harsh environments. A lot of these archaeans are things that show up in super hostile environments, what we would think of as super hostile, like incredibly high temperatures or extremely high concentrations of salt, conditions that would mostly kill all other life forms. Some of the archaeans actually are able to endure it. So for instance, right here, looking at San Francisco Bay, it's a Google Earth image of the Bay Area. And you notice toward the south end of the bay, we've got this area that looks kind of orange. You might look at that and say, what's going on there? Well, if you drill down on that, here's what you get. Looking at it in more detail, we've got all of these areas that are, maybe a mile long, a kilometer or so in size. They're pretty big. And looking even more closely at them, we see that these are drying ponds for salt. This is where there is production producing sea salt because the bay has seawater going in there. And this turns out to be a convenient area in San Francisco Bay where they make sea salt. So you have these drying ponds, and seawater comes in here and then gets cut off from the outside, and under the California sun, the water evaporates and eventually you end up with pure salt. Well, the color doesn't really match with the color of salt in many cases. I mean, here we've got a pond where it does look like salt, but these that are bright orange or red don't look that way. And that's because there are archaeans that are thriving in here as the water's evaporating, and the salt concentration is getting up to, say, 50%. Does anyone have a guess as to how salty seawater is? Like what fraction of salt is in normal seawater?
97.
It's actually about 3.5% salt. So if you take the Pacific Ocean water that's coming into the bay, it's about 3.5% salt. So 96.5% water. Well, here it's now getting more and more concentrated as the water's evaporated, and you get to a pond like this one that's bright red. It's fully colonized by the archaeans, and it may be 50% salt. These archaeans, certain types of them, love the highly salty environments. So there are species of archaeans that thrive under these conditions that would kill everything else. Normally, a high concentration of salt is an excellent way to preserve food. Before we had refrigeration, that was the main way that we preserved fish, for instance. People would catch fish out of the high seas, and they would carry along a lot of salt, and they would carefully have the fish and the salt packed together in the hold of the ship till they got back to shore. That's because the high salt would kill any bacteria trying to rot the fish. But in this case, you've got a species of Archaean that actually loves the salt. So these Archaeans, in many cases, are what we call extremophiles, lovers of extreme environments. Some other Archaeans love super high temperatures, like boiling water, or super acidic conditions, like some of the thermal pools at Yellowstone National Park. So the Archaeans, they have a reputation for being extremophiles, lovers of extreme environments. It's not like every Archaean does that, but a lot of them do. So the Archaeans look a lot like bacteria, but they have some important differences from them, sufficiently different that we put them in their own domain, their own big category. Then we get to the eukaryotes, and we've got a whole section we call a kingdom that's given over to plants. With our taxonomy, we're talking about kingdoms of being big groups within a domain. And the plant kingdom includes, for instance, mosses, ferns, conifers, all flowering plants, for example. This is not including algae. If you think about like seaweeds in the ocean, they go into a different grouping. So this is basically land plants and some other things that you might find in fresh water, but the seaweeds that are in the ocean do not actually fit into the plant kingdom as we define it. There are important differences between those and plants. But examples here of plants, say in the Redwood National Park, for example, ferns, giant redwoods. Animals, right there, you're looking at an animal, believe it or not. Any guess what that is?
Coral?
It's actually a sponge. Yeah, you're close. This is actually a sponge. Sponges are considered to be very simple animals. Then here we've got something commonly known as a sea slug or a nudibranch, and those are more obviously animals. For instance, the sponge just sits there, the nudibranch moves around. Sharks, obviously animals. Sea stars of various types. Any kind of arthropod, like the scorpion, the gorilla, all of these are animals. Well, all animals fit under this heading of what we call kingdom Animalia. Fungi, those are another well-defined group. Fungi have their own unique features. What they will do is they will digest stuff outside of their own bodies, and then they absorb nutrients. So it's kind of like an animal with a stomach, but turned inside out. So a mold like this, instead of having a stomach that it takes food into, it secretes, it exudes digestive enzymes, and they break down, for instance, your strawberries. Lichens are a different arrangement of fungi, very beautiful in their own way. And there are certain really spectacular mushrooms like this one, and some that are nicely edible, like chanterelles. Fungi are another big and diverse group. And if you were to compare them to plants or animals, which one would you think they're probably more similar to? Think they're more similar to plants or to animals?
Animals?
Yeah, we used to not think that. Historically, they've been studied by botanists. Botanists study plants. And people looked at these and they said, well, they grow out of the ground and they just sit there. So, they're just a different kind of plant. But when we look at them more carefully, we realize they're really more similar to animals. One of the key things with that is that they don't make their own food. Plants basically do, and fungi don't. Animals don't. So that's one key thing that makes them more similar to animals. Then we've got this general heading of protist, and you'll encounter this term, and what this is doing is it's looking at all of the different kinds of eukaryotic organisms, and it's subtracting out the plants and the animals and the fungi, and it's looking at everything that's left. Turns out there's a lot left over. And so we've got, for instance, things called slime molds. These are very strange. You've got single-celled predators, like this one that's at the bottom here is attacking another cell. And on the right, this is where one of those prey cells has been eaten. It's going down the hatch right there. So we have the cells here devouring others, being little microscopic predators. We have parasites that move through human blood, for instance. In this case, the parasite is about the size of a red blood cell. It's very tiny. And things like seaweeds in the ocean. These are all different kinds of protists, and they don't really have much in common with each other, except that they are eukaryotes, and none of them are plants, animals, or fungi. What we are left with is sort of this trash can kingdom where we throw everything else that doesn't fit into one of these three groups, because we have a good definition of plants and good definition of animals, good definition of fungi. Then we've got all this other junk left over, and we just say, okay, that's kingdom Protista. It's kind of a fake kingdom. It's basically an association of a whole lot of other life forms that really don't fit under another category. But these are much of the diversity of life on the planet. So we have all this diversity of life, but there's also unity. There are unifying characteristics of life forms. And that unity is useful. Partly we can use it to get a better understanding in medicine. For instance, if you think about all the studies that they do with rats when they're testing maybe some new medical procedure or some new drug, well, rats end up having a pretty similar anatomy to us, and so lab rats end up being a very important thing in medical studies. Before you start doing a procedure on a human, you probably want to test it on mice and rats first. That's because of strong similarities. It's because of this unity of life. We are able to take advantage of this unity, this shared aspect of features of life, to produce medicines, and I'll show you how that works. But as I mentioned, rats have a very similar organ arrangement to us. That's very useful in medical studies. And we are able to produce human insulin using bacteria. Bacteria, we have engineered them now to have the human gene for making human insulin, and those bacteria read that gene the same way that our cells read it, and the bacteria make the same insulin. That's because, again, of this unity of life, shared underlying similarities of life forms. So let's see that. Here, rat. Well, you look at this, you got here, and then this is actually kind of a cartoon, but you've got a heart that's in about the same place as ours. It's flanked by two lungs. You have a trachea coming down to the lungs. You have a diaphragm, which is just a very thin sheet of muscle there. You have the liver, pancreas, stomach, spleen, two kidneys. Only one of them's in view there. Small intestine, large intestine. It's all there. Basically, when you look at this, you find an arrangement of organs that's pretty much identical to ours. So if you're taking A&P1, this is about the first thing you do there. So you will have rats to look forward to in the future. Oh goody. It's not a very nice thing to do, but it is instructive. It's almost uncanny to me looking at this and seeing how virtually identical the layout is. There are some important differences. The lungs are smaller. Humans evolved running a lot after prey. Rats, I don't think, ever evolved to run marathons, so they have small lungs. But the rats do have huge reproductive organs, so one rat can make a lot of new rats in a hurry. So there are some differences, but also strong similarities. That's part of the unity of life right there. The insulin issue. Well, life forms are all using basically the same genetic code, either exactly the same or almost the same. So if you're comparing humans and redwood trees and bacteria, essentially they're reading DNA in the same way. That means that if a human or a redwood tree or a bacterium is reading a little section of DNA, it knows to use that information to make the same protein from it. And essentially, every life form is using the same genetic code. That means the same code book for reading DNA and building a protein from it. It didn't have to be that way, but it is that way. And we think that goes back to all life forms having one shared ancestor. At some point in the very distant past, there was a life form that had this particular genetic code, particular way of deciphering DNA and making proteins. And it evolved eventually into all the life forms we have now, and they all inherited the same code book. So what will happen is that you have DNA, that is genetic code, and you can go through several steps and get to a protein. And essentially, the same DNA will lead to the same protein no matter what organism the DNA is in. There's more to it than that, but that's the general idea here. And what we did, going back to about 1980, is that we developed bacteria that included the human gene for making insulin. And those bacteria read that gene and they make the human insulin, because insulin's a protein. It's encoded in our DNA. And the fact that bacteria read DNA the same way as us means that if they had the human gene for DNA, they could go and then read it the same way and make the same protein. Prior to, say, 1980, does anyone know where we got insulin for diabetics before we had these genetically engineered ones? Well, we actually got it from slaughtered livestock. The thing is, cows, pigs, horses, they all have pancreases just like the rat did. They make their own insulin. It's a little different from human insulin, but it works well enough. And so if you have animals going through slaughterhouses, well, they don't waste anything. And those slaughterhouses would collect huge numbers of pancreases and then use those to get a little bit of animal insulin out. And they purify it as well as they could. And that's what diabetics would use. I know because my dad was diabetic going back into the early 1960s. And we talked about it. He had the insulin he would get from the pharmacy, and it was coming from basically pigs and cows. And then starting around 1980, we had the actual humulin, the human insulin, where we used engineered bacteria that had the human gene put into them. So this is a matter of unity of life. The idea that all life forms are basically using the same genetic code, and this idea of making human insulin from bacteria only works because the bacteria use the same genetic code as us. So that is an underlying sameness of life. Doesn't matter if you're, an Archaean or a fungus or a tree or whatever, you basically are following the same genetic code, either 100% or 99.9%. There are some slight differences, but overall, they're virtually identical. All of that gets to the idea that life forms evolved from a shared ancestor. Evidence that we have now indicates that we had life forms on the planet by about 3.5 billion years ago. Earliest life form showing up around then. How old again do we think the planet is?
4.5 billion years.
About a billion. So within about the first billion, we think that the earliest life forms developed. And one of those became the ancestor of everything that's around now. So it diversified over time, producing these domains with millions of different species and trillions of individuals in them. developing into the diversity of life that we have now. And that original shared ancestor had some features like that genetic code. It had a cell structure. It used proteins. It used DNA and so forth, and passed those characteristics on to its descendants. And over time, we have had descent with modification. We've all descended from that shared ancestor. And what that means is that every life form is related to every other life form. And I know that sometimes bothers people, the idea of being related to other life forms. Personally, I find it kind of inspirational. You're out there and you're surrounded by your relatives, no matter where you go. We think then that the main driver of this has been natural selection, as developed by Darwin in the 1800s. There are other forces involved in evolution, but we think natural selection is the primary one. So that becomes the central theory that we have for explaining why things are the way they are in biology. Natural selection ends up giving our best model for why you have certain things being certain ways. So evolution is this explanatory theory. As we talked about theories are supported by huge amounts of evidence, and they explain and they predict. Evolution does all of those things. So you've got all these different life forms, like seaweeds. What's that? That's known as a naked mole rat. They're hideous, but
is that a pig? A sea pig?
This thing, it's actually a little social mammal. You can see these at the Pacific Science Center. Whenever things reopen, if you're at the Science Center where the Space Needle is, they have a colony of naked mole rats there. And I always like those if I'm in that area. They're just they're hideous, but they're cute. Kind of like some dog breeds, where they're so hideous, they're beautiful. That's the same thing with these guys. But you've got the naked mole rats, you've got paramecium, you've got dragonflies, carnivorous plants, and more. You've got millions of different species, and they're all related to each other. So, if we're talking about evolution, who evolves? Do individuals evolve? Are you evolving right now?
Yes.
Actually, I'd say no. Essentially, you're born with certain genes that your parents gave you, and you basically die with the same genes. Your genes are not changing except if there's some mutation during your lifetime, and that might lead to having a cancer, for instance. But in terms of having genes evolving to make you stronger and better, that's not happening during your lifetime. Generally, you're born and you die with the same genes. All of your cells in your body contain the same genes. So you basically inherit certain genes and you die with the same ones. Individuals, in general, we'd look at them as not evolving. Evolution has to do with changing genes. But populations, the interbreeding groups of individuals going through generations do evolve. And that'll happen over much time usually, sometimes happens fairly quickly. But an example of that would be, for instance, getting antibiotic-resistant bacteria. We have bacteria where we used to be able to kill them with penicillin, and now they're immune to penicillin. Still the same species of bacteria, but those have evolved defenses against the antibiotics. So individual bacteria were killed by the antibiotics, but some others survived because they were naturally a little bit resistant. They became the parents of the next generation. And if you go with wave after wave of penicillin, selected bacteria to be more and more resistant until now, they're pretty hard to kill with penicillin. So we get evolution happening, but basically not within a lifetime. Now, natural selection itself, as I mentioned, that's the primary explanation we have for how evolution happens. And I'm going to show you some of the things involved with this, and then we'll watch a film and probably be done here for today. Life forms normally are producing extra offspring. On average, they're producing more than enough offspring to replace themselves. It might just be a little bit more, or sometimes a lot. If you look at like salmon, you've got 2 salmon coming upstream, and one male, one female, and that female's got thousands of eggs inside. Those all get fertilized, and potentially that's a whole lot of baby salmon. But not all of them are going to survive, right? They produce extra offspring, surplus offspring. Despite the surplus, populations on average stay the same size. So you have competition over resources. There's not enough room in the environment for all of those offspring to grow up. There's not enough food out there for them, and there are threats to them, like from predators. So you'll have competition to survive, and only some of those surplus offspring are going to make it. And there are differences between the offspring, even between siblings, between like 2 brothers. They'll have differences in their genetics. Not all offspring are going to be the same. Some of them have superior genetics, others inferior genetics. The ones with the better genetic arrangements have a better chance of reproducing, and thus you end up with the so-called survival of the fittest. It's a fairly solid, logical thing to look at when you follow your way through that. And here, for example, is what happens with the salmon. Here we've got 2 salmon, male and female, working their way upstream. And she has a whole lot of eggs in there. She's going to lay those eggs, he's going to fertilize them, and perhaps they all hatch. But 5 years from now, on average, how many of these eggs are coming back upstream? On average, how many of these are coming back 5 years from now?
Like 2.
Yeah. If the population stays the same size, then that's what you expect. Nearly all of these die along the way. They might starve, or they might fall prey to disease, or just fall prey to a predator. One way or another, there's a lot of hazards out there. She produced this huge surplus of offspring, and only 2 adults on average will come back up out of all of these.
What?
Well, otherwise the world would be wall-to-wall salmon, but it's not. If she lays thousands of eggs, she's doing the reproduction, which basically ends up replacing herself and her partner. And meanwhile, there are other fish coming up replacing themselves. But it takes a lot of eggs to be able to do that, because most of the eggs will end up dying along the way. So you have differential genes. Some of these are going to have better survival than others. Some of them will have better genetic arrangements than others and have a better chance to survive and make it back upstream, whereas their brothers and sisters don't. So that's part of the logic of natural selection. Here's another one, and this is where I want to show you a short film. This is with a species of mouse that's in the desert southwest of the US, rock pocket mouse. 2 of these mice have a long, happy future ahead of them. 2 do not. Which ones probably have a short, miserable future?
The bottom ones?
Yes, let's say the bottom ones. They are mismatched with the environment. See, these mice have different color patterns, just like people do. I mean, you may get a person with black hair, someone else with blonde hair, still people. Well, that's basically what we have here. We have mice that are light colored, essentially blonde or tan colored, and others that are more charcoal colored. Well, if these happen to be in a place where that gives them good camouflage, they've got a better chance to survive. But this light colored mouse, if he's in a dark background, all of a sudden that's not the way to be. And this charcoal one, if he's in the black rock, great. But if he's on the light rock, again, he stands out. So natural selection, what you can do is predict from it. You can predict that in advance that we're going to see more mice like either the upper left or upper right, and we're not going to see very many mice like the lower left or the lower right. That's a prediction you can make, and you can actually go out and check it. So evolution is testable. That's one example of how it's testable. And I'm going to show you then video where they studied this. It's involving this kind of situation. Okay, that cartoon I just showed you. How many mice were in that picture?
1? Oh.
Yeah. Now they cheated by making these look smaller, but you know the owl is going to see this one first. On the other hand, if the black one was back in these black rocks, he'd have a better survival. And then if these were back in the black rocks, they would die instead. But they actually have done a nice scientific study of this involving measurement, the usual scientific steps. And I want to show that to you here. So it's involving areas in the desert Southwest, including this area that's in New Mexico. That's an area called the Valley of Fire, where I actually went to as part of a vacation where we were going to the caves, Carlsbad Caverns nearby, but we went by the same area that they're going to show in the movie. And you'll see how close a match there is between the environment and the mouse and what has maybe gone into causing that to happen. So let's take a look at that. And this will take me a moment to find. Sorry. Okay, there was a lot of background noise. I just muted all of you. And in general, if you've got anything where you want to comment on, you're able to unmute, but otherwise I'll typically have you muted so we don't get background noise. Okay, so the movie here, let's find it. Here it is. And I need to make sure I'm sharing this with you, so let's make sure of that. Okay, you able to see the movie?
Yeah.
Thank you.
Golden deserts dotted by cacti and brush stretched for miles. Yet here in New Mexico's Valley of Fire, the landscape changes dramatically. Patches of black rock interrupt the sand, remnants of volcanic eruptions that occurred about 1,000 years ago. The eruption spewed a river of lava more than 40 miles long across the desert. As the molten rock cooled, it darkened, leaving any creature dependent upon camouflage in serious trouble. In the complex battle of life, one of the constant struggles is between seeing and not being seen, the evolutionary game of hide and seek. And we've come here to the Valley of Fire in New Mexico, a battlefield, to find one of the tiniest soldiers and what it can teach us about how evolution works. On the desert sands, the rock pocket mouse blends in perfectly, its light-colored fur concealing it from predators. But on dark lava, the same fur makes the mouse stand out, attracting the many creatures that see it as food. These mice are the Snickers bar of the desert. They're eaten by foxes and coyotes and rattlesnakes and certainly by owls and maybe even occasionally hawks. And most of those predators are visual predators. So what happened to the pocket mice that found themselves on this new terrain? When I accompanied biologist Michael Nachman onto the lava, it doesn't take long to find out. Oh, this one's closed. Nachman has been collecting mice unharmed in traps. And it's a dark one. It is, yeah. Now, are most of the ones you find up here dark? Almost all. Not only have the mice here evolved to be as dark as the rock, the color change has occurred precisely where it will conceal them from hunters. A bit of a white underbelly, too. That's right. All of the dark ones here in the other lava flows have a white underbelly, and presumably there's no selection for dark on the belly because predators are coming from above. Left to themselves, the mice show no preference for light or dark rocks. It's the predators that have made the difference. The change in color over evolutionary time in the population is driven by predators weeding out the mice that don't match their background. But how did the dark mice arise in the first place? When a black mouse appears in a light population of mice, that is usually going to be due to a new mutation. And those are random and rare events. To fully understand the pocket mouse transformation, Nachman moves from the lava to the lab. He and his team extract DNA from light and dark mice taken from one desert region. The aim? To find one or more genetic mutations that cause dark coloration. A mutation is a change in the chemical letters that make up our genes. It's a copying error that may occur when our cells divide. Mutation seems to mean that something bad has happened. Well, mutations are neither good or bad. Whether they are favored or whether they are rejected or whether they're just neutral depends upon the conditions an organism finds itself. So for the pocket mouse, a mutation that causes a mouse to turn black, that is good if you're living on black rock. It's bad if you're living out in the sandy desert. The light mice are all on the bottom, here. Fur color is a trait controlled by many genes. Turns out to figure out how dark mice evolved, Nachman focuses on how these genes differ in dark and light mice. One by one, the genes prove identical, but at last, something does turn up. The difference between dark and light mice boils down to a difference of 4 chemical letters in a gene called MC1R. Because the gene controls the amount of dark pigment in a mouse's hair follicles, a mouse with these mutations grows dark fur, which gives it an advantage on a dark background. But still, that's one mouse. How would its dark fur spread to a whole population? This lava flow is about 1,000 years old. And so you might wonder, has there been enough time? It's only been 1,000 years. It's a very short period of time for a new mutation to come along and spread so that all of the mice on this lava flow are black, because really they all are. Indeed, such a rapid spread of the mutation may seem unlikely until you do the math. And the reason is that while only one new mouse born in 100,000 may be black, hundreds of thousands of mice are born in any given year. And then those mice that are black have enough advantage that their babies do better and they have more offspring and their offspring have more offspring. And just about a 5% advantage compounded year in and year out can very quickly turn the whole population black, as we see today. If dark color gives mice a 1% competitive advantage, and you start with 1% of the population being dark, in about 1,000 years, 95% of the mice will be dark. If instead, the dark color gives them a 10% advantage, then it only takes 100 years. Thanks to Nachman's mice, science has an example of evolution crystal clear in every detail. What's exciting about this is that we have a system that's very simple ecologically. You have dark rocks and you have light rocks, and you have dark mice and light mice. It couldn't be simpler. We know who the predators are, what the selective force is. We know precisely the genetic basis of what makes the mice have an advantage or a disadvantage depending upon where they live. All the pieces are finally together. It's a perfect illustration of Darwin's process of natural selection. In fact, it's more than that. For Nachman's mice also counter a common misconception that evolution is a random process. Well, there is one random component, and that's the process of mutation. Mutations occur at random throughout our DNA. Every new organism is born with a new set of mutations. But while mutation is random, natural selection is not. Natural selection sorts out the winners and losers. That's really what the whole process of evolution is driven by. But if natural selection is not random, would it produce the same result under the same conditions? It does. And here's proof. Rock pocket mice collected by Nachman from other lava flows in other parts of the Southwest. These are two different black mice, and they each evolved on different lava flows, and the lava flows are hundreds of miles apart, but the genetic changes that made these mice black were different in each case. And what's amazing to me is how similar the black mice are. We didn't know when we started this whether we would find that they were the same genes or different genes, and we were really surprised to find that they were completely different genes, and yet if you look at the mice, they look almost identical. Clearly, there are different genetic ways to make a mouse dark. But once the beneficial mutations appear, natural selection, a non-random part of evolution, can, under very similar conditions, favor very similar adaptations. In effect, each of these lava flows is like rewinding the tape of life and allowing evolution to occur again and again. And in each case, we find the dark mice have evolved. The rock pocket mice show us that evolution can and does repeat itself, and why evolutionary change is never ending. As environments transform, so must the species that inhabit them, adapting and readapting in the great and complex battle of life.
Okay, so nice little video there from the Howard Hughes Medical Institute. We'll be seeing some more materials from them throughout the term. That's just one of several things where they have some rather nice short films on topics that we'll get into. For instance, later on, we'll be looking at sickle cell disease. That's an important one affecting a lot of people in the world. And there's a very interesting story involving evolution of that. So that's something where, again, they have a short film dealing with sickle cell that we will get to. Another one looking at some fish in the Antarctic that don't have any red blood cells. They basically have ice water running through their veins. The blood looks almost transparent and not a trace of red in it. So that is another kind of weird thing that has evolved involving genetic changes, big ones, with those fish. So we'll see a film with that and various other things along the way. So we're getting near the end of what I had for you here today. I thought we might finish early, and we will in a few minutes here. Questions for me, though, regarding that movie or anything else we're getting to before we do just wrap this up?
Mr. Cooper, this is Precious speaking.
Yes.
Yeah, I was looking through Canvas yesterday, and I saw something that is due tomorrow. About finding a partner, how we...
Okay. Which lab section are you enrolled in? Are you in the 6017 or 6019?
Not even sure.
Okay.
I'm not even sure. I didn't print out the syllable, but I have it on mine.
Okay. I don't think there's anything due tomorrow, but there are some things due Friday.
Mm-hmm. Okay.
don't believe I have anything due tomorrow.
Okay.
We'll be doing a lab tomorrow, and there are different parts of that I want people to finish by the end of Friday. And there's a couple of other assignments too, by Friday.
Yeah. And the lab that we're doing tomorrow is lab 1. Are you gonna like send out the tree so we can kind of like specify them? What kind of tree is it, and which one is that one with the name, or how are we gonna?
Wait, which class are you talking about? Aren't you in my Bio 213?
Yeah.
Okay, this is Bio 160.
Oh my goodness. No wonder. I'm seeing some stuff from Bio 211. I was like, that's why I was like, well, and that's okay. I enjoyed the lecture though.
We're doing.
I was like, this doesn't look like. I said, this look like 161 or 211 because something sounds familiar for 211.
Yeah, actually, it's similar to 211.
Yeah.
Look,
damn.
Our class is meeting at 12:45. Okay.
Okay.
All right.
All right. I better get off. Sorry.
All right. These things happen.
Mr. Cooper,
Yes.
I think there's like some error in syllabus module. I tried to find the module, the syllabus file, but I cannot find it.
Okay, let's go and take a look at that. We've got plenty of time to be doing that. So let's go and take a look at our canvas. Okay, so this is what I have for the section 6017, and here's the section 6019. Those are the sections with the Tuesday lab versus Thursday lab. As for the syllabus, it should be right at the top of home. And so
I mean 16, 17.
yeah, so let's take a look at that as well. I can only change one of these at a time. Oh, and I may have something malfunctioning there. So, okay, let's see if I can fix that. It does look like I had a problem there. So I'll see if I can fix that right here on the spot. Everybody needs to have that. It looks like it may have failed to load up correctly. Okay, maybe now we've got it. Anybody in 6017, are you able to look at this and see if it's got the syllabus visible?
I got it.
Okay, good. You know, I had it in here earlier, and then we had the error that turned up yesterday where I had put the wrong time on it for the labs. So I was uploading a corrected version of it, and it looks like something went wrong with the upload. So maybe now we've got it right.
Okay, thank you.
Okay, well, let me know if there's any other issues like that. Okay, otherwise, we've got just a little bit more to do here on the evolution subject, and we're done with this until later in the term as far as, you know, dedicated treatment of evolution. We do have a whole chapter on it toward the end, and we'll be talking about it along the way. Here's part of what came from that study with the pocket mice. They looked actually at 4 different locations across the desert in Arizona and New Mexico. The one that was in the film was the one over here that's at the right. And what they had were situations where, in the New Mexico situation, you had some areas with black rock and some with the light-colored sand and light rock.
Excuse me.
Yes.
We're still looking at the canvas thing.
You're still seeing that?
Yeah.
Okay, I will try that again. I'm sorry.
Thank you.
Okay. How about now?
Better.
Okay, good. Sorry. So you had these 4 locations across the desert southwest, one of them in New Mexico, which is what they were showing in that movie. And remember, you had areas there with light-colored rock, others with dark-colored rock, and there was almost an exact match between the mouse color and the background color, as you would predict from just a simple understanding of natural selection, you'd predict that, and that's exactly what happened. Then there were two other areas in Arizona that just had the light rock, and there the mice were absolutely a match for the light rock. Then another place where it's a mix of environments in western Arizona, where there's some dark rock, some light rock, and it becomes similar to the one in New Mexico, where it's almost a perfect match. But you've probably got some mice wandering back and forth between the environments, and so there's slight mismatches showing up here and there. Anyway, that's part of the study that was done, which they were showing us about in the film. Now, natural selection itself, we can make an analogy with what's known as artificial selection, and this is something that Darwin did. If you go and look at his famous book, Origin of Species, it actually starts with a chapter on artificial selection. He was very interested in what sorts of things people had done by doing controlled breeding. For instance, producing all these breeds of dogs. Dogs, they all go back to wolves. You sometimes hear dog food ads that are talking about that, and it's true. Every dog today has a wolf as an ancestor. Now, we think that dogs were probably domesticated in several different parts of the world, but always it was from the gray wolf, just different gray wolves in different areas. And so starting with the wolf like we have here on the left, I'm happy to say that's one of my pictures from Yellowstone National Park. Then we've got these breeds, and of course many more. For instance, everything's showing up in these dogs underwater calendars. We have hundreds of breeds of dogs, and all of them have a lot of wolf inside of them. Although we have bred them so that over time their diet is different, it doesn't have to be as rich in meat compared to a wolf. If you feed a wolf dog food, it will starve to death. It doesn't get enough protein. So there have been some important genetic changes that have happened with dogs, but we've done this in just thousands of years. We think that the earliest dogs, people were breeding maybe 32,000 years ago, which is before we had agriculture, and we were already starting to get the very earliest kinds of dogs. But that's a blink of an eye compared to the whole time scale that's available through the history of the planet. It only took us, a few thousand years to do that sort of breeding and more. That's artificial selection. That is humans trying to direct the course of evolution, essentially. For instance, maybe you get a dog where out of one litter of puppies, one of them has these black spots, and you think, oh, that's cool, I want to get more of those. And so you concentrate on breeding the ones with the black spots, and you end up with Dalmatians, for example. Or you've got a dog that's especially good at helping you be a shepherd, and you end up breeding a border collie that's especially good at herding sheep and taking directions for a person. Over time, we have bred all these doing artificial selection. And we've done that with a lot of other things. This just looks like a weed, and basically it is. This is a common weed growing around the Mediterranean, Brassica oleracea, but that is the ancestor to all of our cruciferous vegetables. If you're going and buying broccoli or cabbage, Brussels sprouts, they're all derived from this plant. So here we have all these different crops that you can get in the grocery or grow in your garden in a farm field. They are all derivatives of Brassica oleracea. And it only took us a few thousand years to do this breeding. In fact, Brussels sprouts are a new one. We only got those in the 1800s. That was a brand new mutation. So people doing selective breeding with crops have done a tremendous amount of development of them in a very short time, producing genetic changes by controlling breeding. The idea is essentially that natural selection is nature's own way of doing that. And natural selection is not directed the way human farmers have a directed mindset about what they want to grow. Natural selection is happening randomly, but it has much more time. It has millions, even billions of years to operate. And people were able to breed all of these different varieties in a very short time, why couldn't nature do the same thing? That's the basic argument. So this is all something we'll take a look at more later in the term when we are doing our big chapter on evolution. I just wanted to introduce some of these concepts right now. Otherwise, that is essentially what I have for you here today. I can take questions, but if you want to take off, that is fine too. And I'm hoping some days we'll be able to do this. I'll tell you that when we have this summer schedule with the 3-hour meetings, in general, I think we've got a little bit more class time booked than we would normally have during a term with more weeks but shorter lectures. So as long as we can stay on schedule, which we are so far, I have no problem with us quitting early at times. So if you have any questions, I can take those, and otherwise I'll have this open for office hours later and be doing my later class as well. And if you
We're teaching today.
If you want to take off now, that's fine.
I ordered a book and until now I haven't received it. Who do I call for that?
Okay. So why don't we take a look at that? Who's speaking right now?
Josephine Wanjima.
Okay. Why don't we go into Canvas a little bit and see if we can figure something out on that. So, were you able to go into Canvas and go into the sections where, for instance, we have the materials, the McGraw-Hill Connect materials?
Yes, I did.
Okay. Which section are you in?
Right now? Yeah, 6017, yeah.
Okay, yes. So we go into McGraw-Hill Connect, and mine will just take me right on in there, or into this at least. Otherwise, were you able to establish a connection here where you're able to do the labs, for example, that I posted?
Yes, I was.
Okay. So let's go and take a look at what we get into with the Connect section. I'm going to try going to student view because right here, this is what I get as far as linking to the book. Let me go into student view and see what comes up with that. Well, it looks as if we have the textbook over here at the left.
Okay, I can use it online.
Yeah, do you see anything like that?
I haven't gone, I don't even know how to post to go back there.
Okay, well, if you go into the labs through, or the McGraw-Hill content through that link with the McGraw-Hill that was on the left,
okay,
you should get to something that looks similar to this page where it has access to your lab materials and also to the e-text over here from the way this looks to me.
Okay.
If you click that, then e-text should come up. This is not how I've been looking at it, but it looks like it's how you'll see it.
Okay.
The menus for me are different.
All right.
So let's see what we get here. There we go. There's the textbook. Here's the chapters and so forth. So I think that's what you need to do.
Okay, so I was just expecting a hard copy. I did not need to do that.
You're expecting what?
I was expecting a hard copy. I did not need it to do that.
Okay. Well, the bookstore may have hard copies. I haven't been in there to see what they physically have.
Okay.
Card that the bookstore has, that's about $100. That gives you the e-text and access to the lab materials.
Mm-hmm.
They may also have hard copies. That there is the more expensive option that the bookstore offers. That probably has the hard copy for you.
Okay, because when I ordered, I was expecting a hard copy. I ordered 2 books, but I only received 1. So I didn't know what happened to the biology one.
Okay.
Yes.
All right. I hope we'll get it worked out for you.
All right. Thank you.
You're welcome. You're welcome.
I have a question.
Please ask.
Yeah, can we get the book from right here through the canvas, through the connect?
Yes.
Okay.
If you have the card that's about $100 or $120 from the Highline bookstore, that should give you access to all of the lab materials, but also this e-text, which is what I'm sharing on my screen right now. Do you see that?
Yes.
Okay, so that's the e-text. And with that, we go, for instance, with chapter 1, which would be one of the things for you to be reading. Here's the beginning of chapter 1, for example. So that is something that you would, I think, need to be looking at.
Okay.
I have a question. Are you able to go to the Connect Hill? I have a question about the options on the lab. Because there's 2 sections that say the introduction to virtual labs, and I did
I know what you're talking about.
I tried to do both of them, but one of the labs isn't capturing my cursor when I try to do that phase of the lab, but the other one worked. I just want to make sure that I get the credit.
Yeah. Which section are you in?
If you go to the Connect McGraw or McGraw Connect or wherever the virtual lab is.
Do you have the Tuesday or Thursday lab?
Tuesday.
Okay. So right now, this is what shows up with the lab, the assignments as I have them set up here. But I think if we go into the student view for this, I think it will show 2 different listings for that one lab, which is not how I wanted it to be, but somehow it came out that way.
Okay.
So I'm going into student view for the and there we have it, exactly as you're describing.
So that first one up there is the one that worked for me, but when you go down to lab 1, I wasn't able to finish that one. Does that make sense?
Yeah, it does make sense. Sorry, what's your name?
Courtney Sterling.
Okay. Let me see with the grade book here if you got comment on that, because if you did, it's taken care of.
Okay.
So I'm going to go through and take a real quick look and just see what came over into the grade book. Because if you got it done, then you don't have to worry about it. I'm sorry that appeared twice. I don't know why it did. Okay, Sterling?
Yeah.
Yeah, and it doesn't show credit for that. So we need to, and also I think you still need to do the one with the microscope, the 4 microscopes. Not sure about a good answer for you on that at this time. It was something that seemed a little garbled when I was putting this lab together, and I ended up with an extra copy of that one lab listing here.
At the top, it does show me, it does say C report, and it says 3 out of 3, but I just wanted to ask you because I wasn't sure. I couldn't get the other one to load properly to finish it, so.
You mean the one that's under the lab 1 folder?
Yeah, because every time I went to do to add the droplets, my cursor just kept disappearing. I restarted it, I tried it through Chrome, I did it through Internet Explorer, and it just stopped working, so.
I may end up having to give you credit for it manually.
Okay. I can screenshot it and send it to you to show you that I did do it.
Why don't you do that?
Okay.
So I have a question about the lab tomorrow. Do we use the same link to get into the lab?
Yes.
Okay.
What will happen here, let me go and share with you my web screen. Here we are. So tomorrow, this is the canvas for the section that has the lab tomorrow. So are you seeing where it says 6019?
Yes.
Okay. Well, under assignments, you'll want to go to that and try going to this first one, Introduction to Virtual Labs, and for you, it will look different than it does initially for me. I'll change it so it looks like it should look for you. So I need to make a little change here and change this into student view. And then this will be basically the way it should look for you, is that we have this Lab 1 folder, and it'll have these 6 items in there. And what you'll need to do is click and work through each of the steps, and I will take everybody through an introduction to this when we meet tomorrow. So you get there by way of well, basically this is on the McGraw-Hill, but you should not need to log into McGraw-Hill except when you're first setting up the account.
Okay, now I'm kind of confused because I thought I was going to go use the Zoom, right?
Well, you're using Zoom to talk with me, and I'm
okay.
Zoom is for us to talk and for me to show the stuff.
Okay.
But ultimately, you'll need to use your device to go into the McGraw-Hill and do the assignments. And I'll be directing you through this tomorrow. So if you're not quite sure what to do yet, I think we can work it out first thing tomorrow in the lab.
Okay.
Okay, question.
Yes.
So are we having an actual Zoom meeting tomorrow, or are we just doing the
we're doing
lab tutorials?
Yeah, we're doing Zoom tomorrow. I'm going to start it at 8 o'clock, and what I'll be doing is probably taking about 45 minutes showing you some other stuff, and then turn you loose for the rest of the time doing these labs.
Okay.
But Zoom will be for me introducing what we're doing, and then you'll all need to be working on the labs.
Okay.
Okay.
So we'll be working on the labs by ourselves, right?
Basically, what I'll do is have the Zoom meeting broken up into different groups. So you'd be in a group with about 3 other people, and everybody will need to have their own access, probably using their phone or a computer, to be able to get to these assignments. Everyone will need to do the work. But I'll have them connected by Zoom to each other and also to me. So if you have questions, you can talk with each other or talk with me about it.
Okay.
Essentially, like lab partners. So it will start with Zoom tomorrow at 8.
Okay.
Okay.
All right.
All right.
Yeah. Thank you.
All right. Thank you.
I have a question before we go.
Yes.
About the assignment that we have to turn in. Can we write it on? Can we write it with pen and then make it like a photo and upload it?
Yes. Yes.
Okay.
Uploading a photo of it is fine.
Okay.
And
okay.
write it in any color you want.
Okay. Thank you.
Yes, I'm pretty flexible about ways of getting those things. It's just when I had the question about whether it needed to be written on the crossword, I think it does for you to complete it correctly, and also that helps me to grade it because I'm grading a crossword arrangement. But given that, I think you've got more than one way that you can turn it in.
Okay.
Okay, I think I'm going to be turning this on for a little while.
I have kind of like a personal question. So I was on the wait list for the class, and when I went to, like, you added me onto the Canvas course, but when I went to the tuition, it said that I'm still waitlisted and not registered.
You are, as far as that goes. I need to send information to the registrar to let you in. So I got you in Canvas, so you're able to then immediately keep up with the course content. Otherwise, you and anybody else on the wait list, I have to send a note to the registrar to let you in. Normally we would be doing this face-to-face. Give me a form, I would sign the form. Well, I have to instead send them a communication about it.
Okay, so do I just wait for you to do that?
Yes, please. Please just wait a little bit.
Okay. And do you know when you will get that, just so I know like when to pay?
Well, it probably won't be today. I'm sorry, but I'm kind of buried getting things started this term. So it'll be probably tomorrow.
Okay, thank you.
Okay.
I have a question.
Mhm.
Do I need to take appointment for your office hours?
Sorry, I have an appointment.
Yeah, do I need to take appointment for office hours?
No, I'll just be here and I'll have this open again during the office hours, and anyone who wants to drop in and talk can do it.
Okay, thank you.
If you were talking about something personal or something involving your grade, I'd make sure everyone else was in the waiting room.
Mm-hmm.
Okay.